Hi, my name's Cameron Burke, and welcome to this Kaplan MCAT Science Review for Recombinant DNA and Biotechnology. This video is going to introduce you to recombinant DNA and the important role it plays in gene cloning, as well as discuss complementarity and the way complementarity, hybridization, and denaturation play an important role in a variety of different biotechnologies that you're going to be responsible to know on test day. There are two learning goals you should keep in mind during this video. The first is to be able to understand the steps and conditions for basic DNA biotechnology, so you can recognize and solve problems related to those processes on test day. The second is to understand the ways DNA biotechnology has had an impact on modern medicine. This will enable you to answer questions like, what are the benefits of using gene products produced using recombinant DNA? Before we introduce gene cloning, you'll need an understanding of recombinant DNA. Recombinant DNA is a DNA sequence created by combining genes from more than one source that would not normally occur in nature. Combining a human gene with a bacterial plasmid is one way to produce hormones that can be used to treat diseases. This is accomplished by isolating a gene from human DNA, then amplifying it with PCR. After that, it is inserted into a plasmid, and then the host bacteria creates the desired hormone or protein. Recombinant DNA provides a cost-effective way to create safe hormones that are identical to the ones the human body produces, to give to patients who need them, and is a vital component to gene cloning that you could be asked about on the MCAT. Generally, whenever a gene is being cloned, there are 3 steps that have to take place. We'll get into the specifics of each of them shortly, but an overview of the process is that first, a plasmid has to be selected. Then the plasmid and the gene being cloned are treated with a restriction enzyme. Finally, the newly exposed sticky ends of the gene and the plasmid connect. Now that you've been introduced to the big picture, we can talk about the specifics of each individual step of cloning a gene. The first is to choose the right plasmid for the gene being cloned. Of thousands of possible plasmids, some are going to work better for the gene being cloned than others will, so picking the right one is really important. Some common examples include PUC19, PBR322, and Ti plasmid, but you don't need to memorize the specifics for test day. You just need to understand how the process works. Following plasmid selection, the plasmid and the new gene both need to be treated with a restriction enzyme so they can adhere to each other. Restriction enzymes are enzymes bacteria produce that are capable of cutting through nucleic acid strands at a predetermined sequence. Like plasmids, there are many restriction enzymes to choose from, but all restriction enzymes have the same basic job. The spaces in the sequence associated with each enzyme show where it will cut. Once the plasmid and the gene have been treated, the final step can occur. The last step in the gene cloning process is the attachment of the gene to the severed plasmid, creating a complete circle of recombinant DNA that the host bacteria will use to create new gene products. An important factor in human gene segments binding to plasmids is complementarity. Complementarity describes the fraction of nucleotides in one strand of DNA that are bound to their correct base pair on a second strand of DNA. Strands with high complementarity will hybridize, joining together to form a double-stranded helix through double hydrogen bonds between adenine and thymine and triple hydrogen bonds between guanine and cytosine. Complete complementarity occurs when each nucleotide binds with its pair. Partial complementarity occurs when some nucleotide pairs bind correctly, and some nucleotides are matched with the incorrect pair. When no nucleotides are correctly paired, there is no complementarity. In general, the higher the complementarity between 2 strands of DNA, the stronger the hybridization between them will be. The opposite of hybridization is denaturation. While hybridization requires the formation of hydrogen bonds, denaturation requires the breaking of hydrogen bonds. The higher the complementarity between 2 nucleotide strands, the more energy it takes to separate them. Remember that on the MCAT, you should think of increasing heat as increasing energy, so denaturation of completely complementary strands occurs at hot temperatures. Polymerase chain reaction is one of the most important and applicable DNA biotechnologies of the last few decades. PCR allows for a method to produce large quantities of DNA from only 2 small parental strands. This DNA can then be used for a wide variety of purposes, including medicinal and experimental. The 2 parental strands are multiplied exponentially. When you take the MCAT, the formula to remember is that for every n cycles, you produce 2 to the n power strands of DNA. This means that after 30 cycles of PCR, you have more than 1 billion strands of DNA to work with. PCR requires DNA from thermophilic bacteria. One such polymerase is Taq polymerase, which was originally isolated from bacteria that live in volcanic vents on the ocean floor. Parts of the PCR process involve very hot temperatures, and without a thermostable polymerase, the production of new DNA can't succeed. This is an overview of the entire polymerase chain reaction. It looks intimidating, but don't worry. Shortly, we'll go into detail about each of the steps and break it down clearly. But for now, it's important to know the 3 steps that outline the process: denaturation, hybridization, and replication. The first step of PCR, denaturation, is when the original parent strands of DNA are separated from each other. This is where very hot temperatures come in. After being heated to between 94 and 96 degrees Celsius to allow the hydrogen bonds between the parent strands to break, the DNA is cooled to allow the second step to take place. The second step of PCR is primer hybridization or annealing. Both phrases could appear on the MCAT. This is when a primer selects the specific segment of DNA that needs to be replicated and binds to it. Binding can't happen when it is too hot, so the second step is much cooler than the first. After the parent strand primer complex forms, a thermostable polymerase attaches, and the third step can begin. The third and final step of PCR is replication or elongation, where the thermostable polymerase creates new DNA. It needs to be at least 70 degrees Celsius for Taq polymerase to work properly, so this phase happens at relatively high temperatures. These 3 steps repeat to create the needed amount of DNA. The daughter DNA created becomes the parent DNA for the next round. Every time PCR runs, the DNA increases exponentially, ultimately creating a huge amount. While PCR can create DNA, there are other biotechnologies that can help identify DNA. Gel electrophoresis is one such technology. It can be used to figure out how many base pairs long a segment of DNA is. Gel electrophoresis works by moving fragments of DNA through an agarose gel plate. The gel acts like a mesh barrier, so smaller pieces of DNA can move faster than larger ones. The DNA fragments all move forward because of the negatively charged phosphodiester backbone. All of the DNA segments being tested are inserted at point A and move toward point B due to a current running through the agarose gel. The negatively charged phosphodiester backbone goes toward the positive end of the gel, which is connected to the positive terminal of a battery because it is repelled by the negative terminal of the battery. However, the larger segments get stuck in the gel and thus move a shorter distance. At the end of the process, the experimental fragments can be compared to DNA segments of known length, and this allows you to determine the number of base pairs present in your experimental sample. While gel electrophoresis can help determine the size of a DNA fragment, Southern blotting can help test for the presence of a specific fragment of DNA. DNA is blotted with a sheet containing a probe designed to hybridize with the desired DNA. The process starts with bathing the sample DNA in restriction enzymes to break the strand into segments. Gel electrophoresis is used to separate the segments by size. A sheet of nitrocellulose paper is used to blot the DNA segment out of the gel and onto the membrane. A hybridization probe is placed on the membrane to determine if the desired piece of DNA is present in the experimental sample. Many biotechnologies, including Southern blotting, utilize fluorescently labeled DNA. At a molecular level, a hybridization probe binding to experimental DNA during Southern blotting is just like the hybridization of any 2 strands of DNA. During Southern blotting, the nucleotides in the probe are tagged with fluorescent phosphorus, so the probe and any DNA segments that may have attached to it are easy to locate. On the MCAT, questions about hybridization in biotechnology are likely to be asked about through the scope of autoradiography. When fluorescent nucleic acids are covered with photographic film, the radioactive DNA leaves a mark on the film. When sample DNA is compared to the marks of known DNA, like the ones in column A compared to the ones in column B, the kilobase size and the identity of the fragment can be determined. In this particular experiment, the sample contained the 5 kilobase segment, noted by the appearance of a gray band in column A, but not any 3 kilobase or 2 kilobase segments, indicated by the absence of a gray band in those spots in column A. Now that you've been introduced to the content for this science review, you can try some practice questions to check your progress with the learning goals. Practice question 1 addresses the benefits of gene cloning. The correct answer is D. Recombinant forms of hormones are cheaper, safer, and more pure than hormones from other sources. Choice C is correct. If 2 strands of DNA aren't completely complementary, they will require less energy to separate. Additionally, on test day, keep in mind that if 2 answers have the same meaning, as in the case of choices A and B, both must be wrong. In this example, because adenine and thymine pairs and guanine and cytosine pairs are the only ones that can make up bonded nucleotides in DNA, if there is a high percentage of GC pairs, that means that there has to be a low percentage of AT pairs. For this reason, you can eliminate both A and B. The final practice question gives you some experience troubleshooting biotechnology processes. D is the correct answer. PCR needs the right balance of substrates, enzymes, primers, polymerases, and time to produce large amounts of high-quality DNA. Although it was correct on this particular instance, on test day, all of the above is not more likely to be correct than any other answer choice, so be cautious when considering that option. To recap, recombinant DNA is a compilation of nucleic acids from more than one source. It is useful for producing human genes isolated from a chromosome, and then amplified using PCR, to be inserted into a bacterial plasmid so the bacterial host can create hormones. Complementarity is the degree to which two DNA strands complement each other and affects hybridization and denaturation. The higher the degree of complementarity between two strands of DNA, the more energy it will take to separate them. Recombinant DNA and hybridization both play important roles in biotechnology. PCR increases the amount of DNA through denaturation, hybridization, and elongation. Gel electrophoresis can help identify the length of a DNA fragment, while southern blotting can identify unknown DNA fragments using hybridization. Thanks so much for watching this Kaplan MCAT Science Review for Recombinant DNA and Biotechnology, and good luck when you take the MCAT.